We have introduced now the two important proteins involved in the circulatory system, that is the transport of oxygen. And we said that hemoglobin, heme by the way means blood, is oxygen transport in blood as a function, and myoglobin, myo means muscle, is oxygen storage in muscles. Let's be a little more specific about that. When hemoglobin and blood pass through tissues and muscles, the oxygen is going to equilibrate between the blood and the myoglobin. And myoglobin's job is to grab as much oxygen as it can from the blood. I like to call it an oxygen scavenger. It binds O2 very tightly all the time. Your muscles want to get as much oxygen as they can because it's needed for respiration. And it basically has this protein, myoglobin, that is super sticky for oxygen, has a very high affinity for oxygen. So all we need to do to describe how the structure of myoglobin meets its function is to show how it binds oxygen very tightly all the time. It's a pretty simple function. Now, unfortunately, it's not quite as simple as we've portrayed it to be in earlier videos with regard to structure. In fact, myoglobin, which we have shown over here on the right, this is the myoglobin monomer. It's one protein chain that folds up kind of like an egg shape. It's all alpha helix and loops, no beta sheets. For all that structure that we talked about for myoglobin, it can't bind oxygen at all on its own. It needs an extra help group or help molecule that's going to bind to it. And that molecule that helps it out is called heme. And heme is an extremely important molecule through all of biology, so let's take a little moment to talk about what it is. There's some terminology we need to cover. So heme is shown right here. It's a big flat molecule with aromatic rings that have nitrogens in them, and it also has some carboxylates out here. So it's got a very nonpolar area and a polar area over here. It's kind of almost amphipathic. That part is called the porphyrin ring, all the organic chemistry. And porphyrins are found in a lot of molecules in biology, in chlorophyll and vitamin B12 and things like that. Also in heme, though, besides the porphyrin, is an iron +2 atom. That's iron 2. Has to be iron 2 to function in this capacity in myoglobin. Now, heme is an example of what we call a prosthetic group. Think about where you've heard that term before. A prosthetic limb is an extra limb that you attach to somebody who is missing a particular function. Let's say they're missing the ability to walk. You'd give them a prosthetic leg. In this case, we call this a prosthetic group because on its own, the protein part of myoglobin can't bind oxygen at all. And since that's its function, it's really not going to be able to do what it needs to do. So we add this prosthetic group, and what you can see is that it slides right down in the interior of myoglobin. Maybe you can't see that, but it's going to be, the nonpolar portion is going to slide right into the center where the nonpolar amino acids are. And there's the iron right there. That's where oxygen is going to bind. In fact, we can show that to you. We'll take an oxygen and say that if it were to encounter a myoglobin, it would migrate its way in. Oxygen is diatomic O double bond O there, and it would go in and bind to the iron. I'll show you better three-dimensional pictures of this in class. Now, more terminology for us here though. If you have myoglobin protein on its own, it is called apomyoglobin. That's a general protein term. Apo means a protein that's missing some extra functionality about it. And apomyoglobin without the heme is non-functional. It cannot do what it needs to do because it doesn't have that iron and that heme. We use the term holo, H-O-L-O, myoglobin, for a protein that includes the prosthetic group, and that is functional. So let's make a note of this all. Myoglobin alone, non-functional apomyoglobin. Myoglobin plus heme is going to be a functional holomyoglobin. And that's terms that we may use in other contexts in this class as apoproteins and holoproteins. So once we have myoglobin with a heme and particularly an iron in it, it is now free to bind oxygen. Let's go down to the bottom of the page, and before we move on to the next video, I want to just give you a little chemical formula here for the way we think about this. When you have myoglobin, that is holomyoglobin, and it encounters an oxygen, it can undergo a reversible binding reaction like this. We write down myoglobin dot O2, and this is now a complex where the O2 and the myoglobin are together. So this can go in the forward direction with oxygen binding, and it can go in the reverse direction. Now I can tell you that the forward direction is much better than the reverse direction. That's because myoglobin and oxygen are very sticky to each other, and they tend to favor the complex between them. This is it. Myoglobin can bind one oxygen because there's only one heme, one iron, and therefore one oxygen bound. It's a very simple situation, and we're going to see how that's quite different for hemoglobin later. Let's move on to the next video where we're going to talk about how we can do this graphically and mathematically.